So welcome back students. We will just continue with the components of the barcoding project. So the first component, it is a specimen. Then the second component, it is a laboratory analysis. Then the third component, it is a databases, and the fourth one, it is a data analysis. And we discussed the first and the second and almost a third in the previous video lecture. We will just continue with the data bases. The two main databases that are present, which is available for all, they are the International Nucleotide Sequence Barcode Collaborative, and it is a Barcode of Life database. So these are the two. So once again, the first one, it is the International Nucleotide Sequence Database Collaborative, and the second one, it is a Barcode of Life database. So these are the two different databases which are available globally. Then the last one, the fourth one, it is a database. That is a fourth component. The specimens are identified by finding the closed matching references in the database. You'll be getting the sequences and you'll be just comparing the sequences available. And this data analysis working group of COBOL, that is a consortium for the barcode of life, has created the barcode of life data portal, which offers the researchers facilities to store, manage and analyze, and also to display the barcode data. So all these things we are able to do in this database. You just have to log in and you can just add to it. Okay, so it is a website accessible for the DNA barcoding. So that's all the procedure, and we will just see. So this is a barcoding, color barcode, which is available. Then you can see these are the four bases available, nucleotide bases, and each basis, the adenine gives a green color in the sequence, thymine gives a red color, cytosine gives a blue, and guanine gives a black. And these are the few examples for the barcodes available. And this is the barcode for chimpanzee. Then this is a sequencer. It is automated sequencer, DNA sequencer, and this is the PCR which is available in the laboratory. Then in future, this type of barcoder will be available, in which we will be able to feed a small part of the specimen, and automatically you will be getting the barcode, and we will be getting all the details from this barcode, as if we are just loading the blood sample to get the glucose level, etc. Likewise, a barcoder device will be developed in future, and we will be getting the data in few seconds, and the links for the database references you will be getting, and you will be getting a taxonomic GPS reusable by all the non-specialists. So any individual can use this type of barcode to identify the different specimens. So these are the few points. Then we have just discussed about the advantages, but still the barcoding is very much important under the when we look into the conservation strategies. We are able to protect the endangered species, and we are able to conserve the different species. We are able to track the adulterations, then identifying the agricultural pest. Water quality can be tested by using this type of barcoding techniques. And we are able to identify the all stages of life, that is egg, larvae, nymph, pupa, adult, etc. Even from the fragments, we are able to identify the organism. Then from the stomach contents, from the trace of the ecological food chain, we are able to identify the specimens. We are able to control the food, customs control, invasive species control, disease vector control, and we can use in the forensic, then forestry, agriculture, education, in almost all the fields of science we can use this barcoding technique for various processes. Then these are the strength and the weakness of barcoding techniques. The strength are it is that it is an alternative tool for identification, by which we are able to identify the new species. It works on all forms, and it unveils the undescribed species. So these are the strength. But still there are the few weakness. There is no universal DNA barcode region. That is, if you are going for an animal, we can use the CO1, but for the plants it is different. Then it is difficult to resolve the recently diverged species. Then it identifies the interspecific genetic variations only, and it is a single approach. So these are the few weaknesses, but still it is having more advantages than disadvantages. So it's very important. And to conclude, the DNA barcoding has emerged and established itself as an important tool for the species, mainly for the identification and the phylogenetic studies. So it's very important in the identification process and to study the phylogenetic process or the evolutionary, to trace evolutionary history. This type of barcoding technique is very much used. Then it has been useful for protecting the endangered species, for identifying the agricultural pests and the disease vectors, for tackling the adulteration in various products, and also in sustaining the environment. And the various case studies are also there. Dispute identity of organism has been resolved by using this type of DNA barcoding techniques. These are the few examples, just for your information. Both in the case of the plants and animals, there's a type of butterfly dispute was resolved. Then philanthus is a plant species. These are the something, and mainly we are able to conserve our ecosystem. So this is where we stand today. So on a top, and you can see only a small it is identified species, and the larger part of the iceberg which is embedded in the ocean has to be identified. So only a bit has been identified. So this DNA barcode has its own importance in the conservation strategies. So you can just revise this part, that you have to study what is DNA barcoding. Then you have to study a briefing of DNA barcoding procedure, which are the components in DNA barcoding, applications of DNA barcoding. Then applications is one of the major important topics that you have to study for the examination point of view. Okay, that's all in this video lecture. Thank you.